Welcome all. It's my immense pleasure and privilege to convey my sincere gratitude to ScienceSoar Publications providing this opportunity to deliver a talk on biosensor for human perspective. What is biosensor? It is a device that uses specific biochemical reactions mediated by isolated enzymes, immunosystems, tissues, organelles, or whole cells to detect chemical compounds, usually by electrical, thermal, or optical signals. The term is also defined as a sensor which incorporating biological elements such as enzymes, antibodies, receptors, proteins, nucleic acids, cells, or tissue sections as a recognition element coupled to a transducer. Let us see the principle of the biosensor. It contains a sample which recognize the stimuli by the biorecognition elements and followed by the transducer which provides the signal processing devices. It provides the biological materials are immobilized and a contact is made between that immobilized biological material and a transducer. The analyte binds to the biological material to form a bound analyte, which in turn produces the electronic responses that can be measured. Sometimes the analyte is converted to a product which could be associated with that release of heat, gas, electrons, or hydrogen ions. Then the transducer converts the product linked changes into electric signals which can be amplified and measured. Here you can see the diagrammatic representation of these working principles. The biosensor basically involve the quantitative analysis of various substances by converting their biological actions into measurable signals. Generally, the performance of the biosensor is mostly depends on the specificity and the sensitivity of the biological reactions, besides the stability of the enzymes. The analyte which diffuses from the solution into the surface of the biosensor, the analyte reacts specifically and efficiently with the biological component of the biosensor. This reaction changes the biochemical properties of the transducer surface. This leads to a change in the optical or electronic properties of the transducer surfaces. The changes in the optical or electronic properties is measured or converted into the electrical signal which is detected. This is also a example of biosensor. Here the sample or the standard which are coupled with the receptor and followed by a transducer and followed by the amplification of the signal treatment. We can use microorganisms on the surface. It is also coupled with the receptors and it is also conduct the signal to the transducer followed and reach to the amplifications. You can see by giving the arrow. Yeah, we get the signal in the form of the electrical or physicochemical properties. What are the basic biosensor components? First one is the analyte, biological elements, transducer, processor and monitor. We can use the biological elements or enzymes, nucleic acids or any receptors which can be bind with the substrate and it is activation of the enzyme and which gives the signal. What is analyte? It is a molecule in the form of protein, toxins, peptides, vitamins, sugar, or metal ions. So many, for example, given here the cholera toxins or the glucose molecules are the typical example for the analytes. Then biological elements, elements in the form of antibody, enzymes, cells, polymer or hydrogels which contain the receptors. It has a specific affinity with the binding of the compounds. Transducer, the transducer or the detector element which transform the signal resulting from the interaction of the analyte with the biological recognition elements into the another signal that can be more easily measured and quantified. The amplifier, microprocessor and the display. The biosensor reader device with the associated electronics or signal processor that are primarily responsible for the display of the results. Here you can see the function of this biosensor. The sample, it can be integrated with the bioreceptors and which are connected with the transducer in the form of electrodes or transistor, thermistors, optical fibers, piezoelectric catalyst. It gets a signal of the electron changes or conformational changes or physical chemical changes, and it is the signal is transferred to the amplifier in the form of electrical stimuli, and it go for this data processing will be obtained in the form of microelectronics. For example, we are routinely using this biosensor for the pregnancy test for the detection of this human chorionic gonadotrophic hormone protein in the urine sample. Then we have to use the glucose monitor devices for detection of this diabetics, monitor the glucose level in the blood. Then infectious disease biosensor from RBCs. Here we have the research center. We are also working on this biosensor devices establishment for the extraction and isolation of the pigments from the various biological samples and development of photovoltaic cells for dye sensitized solar cells, and other research centers are also available in our institutes. We are working for in various aspects. For the isolation of the pigments, for example, here we are given for spirulina platensis. We have a large scale production unit for around 30,000 liter capacity of this spirulina cultivation unit. Then we collect the spirulina species and allow to dry the collected samples and make this powder and go for the extraction of the pigment extraction by different solvent procedures. We are also worked with this and the plant with the pigments, especially Bauhinia purpura plants, and also use the microorganisms. Serratia marcescens is a wonderful organisms, and halophilic organism also used for this isolation of protein. It is a typical experimental setup for preparation of dye sensitized solar cells. For preparation, we have to coat these pigments with the titanium dioxides as a electrode. First, we clean the conductive fluorine doped with the tin oxide FTO glass plates. Then identify the conductivity side. Then with the conductive side facing up, apply two parallel strips on tape on the two edges on the glass plates. The titanium oxide paste was prepared with the 0.2 gram of commercially titanium oxide and 0.1 molar of nitric acid solution and 0.08 gram of polyethylene glycol were added. Then apply a portion of paste near the top edge of the FTO glass plate by the glass rod and spread the paste across the plate. The titanium dioxide acts as a photoanode prepared by the detector blade technique. Then titanium oxide film was air dried and heated at 500 degrees centigrade in a furnace for 45 minutes. Then prepared titanium dioxide film was immersed overnight in natural pigments and rinsed with ethanol and finally air dried. Then assembling of the photovoltaic cells. The dye-sensitized cells was assembled by using dye-stained titanium dioxide photoanode on act as a counter electrode facing each other with two binder clips which used to hold the electrodes jointly. The iodine electrolyte solution was poured on the edges of the plate and allowed to disperse between the plate by capillary action. This resulted in the formation of the sandwich type cell. It is you have to see the analysis of the photovoltaic properties. The photovoltaic performance of dye sensitized solar cell using Serratia Morrison strains and Spirulina platensis and Bagheera purpura plant extract as a natural dye as photosensitizer was determined by recording the current potential curves using the visible illumination and which are displayed in the picture. The conversion efficiency of dye sensitive solar cells which exhibited open circuit voltage, we obtained the 0.092 volt the short circuit current density of 1.6 milliohms and the efficiency of 1.2%. We work in this field and the generation of various dye sensitive solar cells and made good publication in different reputed indexed journals. Thank you for patient listening of this biosensor applications for human perspectives. Once again, I convey my sincere gratitude to the Science Shore for providing this wonderful platform for exploring science to the human community. Thank you.